Okay, welcome to everyone who's joined us so far. Welcome to Ocean Acidification Week 2024. We'll just wait for a few more people to arrive and we'll get started in just a moment. Okay, thanks everyone for joining us for Ocean Acidification Week 2024. We will just get started in a second. Maisie, I can see that you've raised your hand. Can I help you?
Sorry, that was by accident.
Oh, no worries. Okay, perfect. Okay, great. Well, welcome to Ocean Acidification Week. Um, my name is Rachel Hale. I'm a marine biologist based at NIWA in Aotearoa, New Zealand. I'm the coordinator for this session, where we'll be presenting work from the Pacific Islands and Territories Hub. So please note that this session is being recorded. Ocean Acidification Week is presented by 4 sponsoring organizations. First, GOA-ON, the Global Ocean Acidification Observing Network. Second is NOAA, the United States National Oceanic and Atmospheric Administration. The third is the IAEA OA-ICC, the International Atomic Energy Agency Ocean Acidification International Coordination Center. And last but not least, the IOC UNESCO, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific and Cultural Organization. A bit of background about GOA-ON before we start the presentations. So GOA-ON was established in 2012 with just a handful of members. Since then, GOA-ON has grown immensely. It now has over 1,200 members from 114 countries. GOA-ON also consists of 10 regional hubs, which span across continents and oceanographic regions. And we'll be hearing from most of these hubs throughout Ocean Acidification Week. If you're not a member yet, you can join GOA-ON today by visiting goaon.org. Ocean Acidification Week debuted in 2020 and returned in 2021 when events and conferences were postponed due to COVID-19. Following the successful in-person symposium on the ocean in a high CO2 world in 2022 and another successful Ocean Acidification Week in 2023, GOA-ON is bringing back Ocean Acidification Week for 2024 to maintain momentum around OA research and provide a virtual platform for the ocean acidification community to exchange their latest findings. We are thrilled to present a wide range of ocean acidification topics and speakers from around the world. During the presentation, all participants are in listen-only mode. You are welcome to type any questions into the questions box, which can be found at the bottom of the control panel on the right-hand side of your screen. We'll be monitoring incoming questions and we'll pose them to our speakers during the question and answer session, which will be at the end of the final presentation. For the discussion, you can also use the raise hand function, also in the toolbox at the bottom of your screen, and we will call on you to ask your question directly. So with this, I would like to introduce you to your moderator for this PI-TOA Hub session. The moderator is Luia Taise. Luia is a senior lecturer at the National University of Samoa. She is a principal scientist for NUS's Ocean Acidification Research Program. She did her PhD thesis on the impacts of climate change on the green seaweed Kaulerpa, focusing on the effects of ocean acidification temperature and light on the physiological responses of Caulerpa species. Luia, the floor is yours.
Hi, everyone. Thank you, Rachel, for the introduction, and to our audience for tuning in around the globe. This session is the Pacific Islands and Territories Hub session. Increasing atmospheric CO2 levels and its implications on marine organisms rise the need for a multi-scale approach to ocean acidification research and monitoring. In this session, we look into processes and environmental factors that affect marine species in the context of ocean acidification, mitigation approaches, and monitoring activities in the Pacific region. Each speaker will provide a 12 to 15-minute presentation, and we will hold all questions until the end for a 30-minute discussion as a group. With that, we can begin with our first speaker. I would like to introduce Sebastian van der Velde. Sebastian is the head of the National Marine Carbonate Chemistry Facility of Niue, New Zealand, and a lecturer in marine chemistry at the University of Otago in New Zealand. Focusing on the biogeochemical cycles of carbon, iron, and sulfur, Sebastian studies the past and present ocean using a research approach that combines fieldwork and laboratory incubations with numerical modeling at the local, regional, and global scales. Specific topics are the importance of burrowing fauna for cycling of carbon, iron, and sulfur in the Earth system, spatial patterns of redox conditions and nutrient availability in low oxygen oceans, and the impact of anthropogenic sediment disturbances on the coastal carbon cycle. Through this work, Sebastian aims to improve our understanding of the Earth's evolution and to provide new insights into the links between life and its geochemical environment. With that, Sebastian, the floor is yours.
Thank you, and thank you for this introduction. So I'll start, and I hope there's not too many gray bars on my screen. And see if we can get this going. So how does this look? Can you see my presentation?
Yeah, it's looking good.
Yes.
Okay, excellent, good. All right, so yeah, I'm the first speaker, and well, first of all, thank you for giving me the time to give this presentation. And I'm immediately starting by probably taking it away from ocean acidification and talk more about ocean alkalinity or ocean alkalinity enhancement, because I think this is something where a lot of groups and industry are moving towards, but also something where the ocean acidification community has a very strong grounding, has done a lot of work over the last decades that could actually be used in this shift. And to immediately jump into it. So I don't have to explain this to this audience, but you know we've come to the stage where most of the CO2 or the mitigation trying to stop emitting CO2 has been a bit too little, and we're reaching the too little too late stage. So if you want to keep the increase in temperatures and the increase and the negative effects of climate change within acceptable limits, we're gonna not only have to stop emitting CO2, but we'll also have to start actively removing CO2 from the atmosphere. And so there is a need. This is not a little bit, this will be gigatons of CO2 that we will have to remove from the atmosphere. And so one of the ways we could do this, we could actually use the ocean. We could use the ocean as a CDR or a carbon dioxide removal technology. And so to get a bit of the basics on how this would work. So we have within the ocean, there's an equilibrium within seawater. There's an equilibrium between the amount of dissolved CO2 or the amount of dissolved inorganic carbon that's present, the amount of alkalinity that is present, and the eventual pH and the eventual partial pressure of CO2, of dissolved CO2 in the water phase. And so what we could do, we could imagine if we increase the partial pressure of CO2, which is what we're doing now by increasing atmospheric CO2, we're moving to the right on the scale, which means we're increasing the amount of DIC. So there's more CO2 total dissolved CO2 in the ocean. And at the same time, we are decreasing pH, right? This is the classical ocean acidification problem. This is what most of us are studying. We could also turn this around. We could, instead of increasing the PCO2, we could increase the alkalinity. Now, what happens if we increase the alkalinity is, on one hand, we are increasing the pH, but on the other hand, we're also shifting this equilibrium, this carbonate equilibrium, away from the CO2 phase and into the bicarbonate and the carbonate ion forms, which means that we're actually decreasing the amount of or the partial pressure of CO2 in our seawater parcel or in solution. And because the ocean is an open system, this will find the re-equilibration. So there'll be the PCO2 will normalize itself and take on again the PCO2 of the atmosphere. So what happens then is that we stay on this 400 ppm isobar. And as we're increasing alkalinity, we are doing two things. We're increasing the amount of DIC, which means that there is CO2 from the atmosphere that is becoming dissolved in the ocean. So the CO2 removed from the atmosphere into the ocean. And we're also increasing the pH. So we're actually counteracting the effect of ocean acidification. And so this concept, this is essentially the concept of ocean alkalinity enhancement, right? If we can increase the alkalinity, we can increase oceanic CO2 uptake, and we can also, to a degree, counteract ocean acidification. And so there's been a whole spread of different technologies that have been proposed or approaches, but you can really group them into two categories. One of them is focusing on the natural weathering process. So if we have silicates or carbonates that get weathered on land, they create alkalinity, and that ends up in the ocean. Trying to stimulate this. So this is the more rock-based or the mineral-based approach, where we're using silicates or carbonates, putting them in environments that weather very rapidly and increase the alkalinity supply in that way. And a second one is a bit more electrochemically approach, where you're actually splitting water, you're splitting it in acid and base, and you're taking away the acid and putting the base back into the ocean. Now, all of these technologies, it doesn't really matter which one, they all have the same needs or the same problems associated with the readily applying these in globally. And I'm leaving aside here any potential environmental impacts of releasing minerals in the environment, but I'm purely focusing on the chemical methodological problems. And one of them is that there are natural sources of alkalinity, there are natural weathering processes, there is carbonate dissolution in sediments, there's other processes that generate alkalinity. If we are gonna artificially increase alkalinity in any spot in the ocean, there will be feedbacks that we need to account for. So we need to account for any loss of alkalinity through any potential links between enhancing alkalinity, adding alkalinity to the ocean, and somewhere else a natural source that decreases. And this has been coined or named the additionality problem. To give you a little bit of an idea of what this problem actually entails, I'll just show you a little example of experiments we've been doing when I was still in Belgium, in Antwerp. And essentially what we were doing is we were incubating about 1 square meter of seafloor. And over like 10 years, it was a very long running experiment. And we added olivine, which is a silicate mineral that weathers very fast. And as it weathers, it releases alkalinity. And we've added olivine to some of these tanks. And so what we did, we had several treatments. So we had the treatments where we have no addition. We have the treatments with olivine addition. And then we also had the treatments where we have bioturbators or natural communities of worms living in the sediment. And then also the treatments without any bioturbators. And so we were checking the alkalinity release or the enhanced alkalinity release through olivine addition. And so what we found, the first thing was, if we looked at the tanks where there were no natural community of bioturbators and we added olivine, we could see that there was an increase in alkalinity release, which meant that the addition of olivine enhanced the weathering, increased the alkalinity release, so it would be a viable ocean alkalinity enhancement technology. When we added olivine to the tanks with head bioturbators, we did see no decrease. We had the same amount of alkalinity release in the tanks with and without olivine. However, what we did see is that when we looked at the olivine grains within the sediment, is that we had very clear evidence that these grains were shrinking. So they were dissolving. So they were releasing their alkalinity. We weren't able to catch it or capture it in, well, there was no alkalinity coming out associated with the dissolution. So the question arose like, well, where does the alkalinity go, or why don't we find it? And so we did a bit of mass balance modeling. And actually what we did find was that as you had no biturbation or no animal community, there was no carbonate dissolution, which meant that if you added olivine, you had alkalinity coming from the olivine, and that would leave the sediment. So you ended up with an enhanced alkalinity release. But if you added, if you had biturbation in your tanks, you stimulated carbonate dissolution within the sediment. Now, carbonate dissolution is bound to the saturation state. As your saturation state goes lower, there's more carbonate being dissolved. So what actually happened was that we added the olivine. The olivine dissolved, released alkalinity, but it shifted the saturation state of the carbonate. So it increased that saturation state, which actually inhibited dissolution. So we didn't have an increase in alkalinity release. We did weather the olivine, but we inhibited the amount of carbonate dissolution. So there was no net gain of alkalinity. So this is just a bit of an extreme example maybe, but just to show that we can add alkalinity to the ocean, but we might inhibit or lose alkalinity on the other side. A second problem is that we need to be able to quantify how much alkalinity we add, or conversely, how much CO2 we capture. And so this is what's related to the monitoring, reporting, and verification side of OAE. And this becomes a problem of small changes against a big background, because we have a lot of alkalinity already in the ocean. If we're adding alkalinity, this will always be in very small amounts. And so we should be able to, against the background, which is both large and very variable. There's a seasonal variability depending on there's a lot of variability both on daily scales, yearly scales, seasonal scales. And so to capture a small addition against such a background is very complicated. And one of the ways around this is that you could use the salinity-alkalinity relation, which is locally or regionally changing. But so if you have this salinity-alkalinity relationship, what you could do is you could look at after your addition if there's a shift in this relationship, which then could indicate or could help you to quantify how much alkalinity you have added. So for both of these problems, either we need a lot of new material, need to go out, measure a lot of new things, do a lot of new experiments, or we could tackle this with existing time series. So if we have the carbonate system parameters, alkalinity, DIC, pH, over long time scales and a lot of data, we can actually use that data to try and narrow down or address these two different problems. And so this is sort of the point I wanted to make, is that actually the ocean acidification monitoring of the last few decades has actually created these databases and has a lot of value for moving forward into the ocean alkalinity enhancement or CDR space. And so I kind of see this as a growing knowledge base based on everything as a community we have been doing and will be doing. Whereas in the early 90s, we started with the studying the carbon cycle to look at how much anthropogenic CO2 has been taken up. We then moved with the realization that ocean acidification had a large impact to the more monitoring, ocean acidification monitoring that's been going on within this community. And so now we could take the next step towards more ocean-based solutions rather than from the monitoring, we could actually try and action and try to mitigate climate change by actively using the knowledge we have gathered. And so that's where I'll end. So just a few take-home messages. OA I still believe, has versatile. It provides versatile options, and it is something that could work for CDR, provided we take the environmental impact and the additionality into account. Accounting for these natural processes and this variability requires a good understanding of the local systems, and so this is where actually the acidification, ocean acidification monitoring has been going is very priceless. And it's something that as a community we could use. And And as I generally end every presentation talking about CDR or carbon dioxide removal, it's still the reducing of the CO2 emissions that will be critical, that we will need to achieve climate stabilization. All the CDR is only going to help us give the little last push, but it's the reducing of the emissions that is going to have to make the big step that's going to be needed. So with that, I'll just say thank you also to the research group I worked with before I came down to New Zealand. And I would, I guess questions was for the end of the session. So I'll stop sharing here and give it back to the moderator. Thank you.
Thank you so much, Sebastian, for that enlightening and technical presentation. That was very interesting, and we hold all questions toward the end of the session. Next, I would like to introduce our next speaker, Melissa Melendez. Melissa is a postdoctoral researcher at the University of Hawaii at Mānoa, working on an NSF project that aims to develop a high-frequency autonomous dual sensor to measure seawater total alkalinity and pH to evaluate metabolic processes in Hawaiian coral reef areas from in situ time series or mooring and process study or drifters data. Melissa also analyzes data from different CO2 buoys in the Atlantic and Pacific on the first comparative study that aims to provide a more holistic view of how coral reef ecosystems are being affected by ocean acidification. Melissa, thank you. The floor is yours.
Thank you for the introduction. Let me share my screen. Oops. And can you see that?
Yep, we can see it. Oh, it's in
Okay.
presenter mode.
It is? Oh, it is in presenting mode, or can you see the...
It's the presenter screen, not the full slide.
Oh, let me check that. Better?
Perfect.
Okay, good. All right, so thank you so much for the introduction. I apologize for my dog that is barking next to me. But as I mentioned, I work with a bunch of collaborators from here, from the University of Hawaii, including Chris Sabine, Lutzinor, Adrian Suchan from NOAA Pacific Marine Environmental Laboratories. Thank you so much for the invitation. I have many other collaborators and students that help with this project. I'm just going to present a little bit. As you may all be aware, coastal coral reef ecosystems are critical in the nearshore regions, and they show distinct and particular carbon chemistry dynamics compared to the open ocean. And also ocean acidification manifests quite uniquely in these nearshore reef areas because of those natural processes. There is also fine-scale fissures often overlooked during the models and global models that cannot capture these variations, and they focus on long-term and seasonal cycles. And to constrain this carbon chemistry dynamics across different time scales and spatial scales is particularly challenging in these nearshore areas because of the lack of data and because all of the complexity that the coastal areas processes have. So our study and what I'm going to present today is focusing on understanding the diurnal changes in carbon chemistry and identifying the dominant processes shaping these fluctuations. I'm using different measures and tools, but we are focusing on understanding or analyzing the data from this map CO2 buoys. We're fortunate that we have two here in Hawaii. They measure carbon chemistry at every 3 hours, and they measure seawater temperature, salinity. Some of them has oxygen, pH. They also have CO2 in the air and in the water. And they look at something like that. They are provided by NOAA and the Ocean Acidification Program, and we are in collaboration with them. So as I mentioned, we have two buoys located in Oahu. They are located in the east side of Oahu and Kaneohe Bay. And I'm showing here just a quick kind of overview of where they are located relative to the shore. Our nearshore buoy, the Reef buoy, is located around 1.2 kilometers from shore, and it's in a reef area where the dominant kind of currents pass through all of this beautiful reef, and the buoy kind of like capture all of that reef dynamics. We have the offshore buoy. It's a little bit, it's 3 kilometers from that reef buoy and around 5 kilometers from shore. And that represents more like open ocean dynamics and more like blue waters and members. So we want to understand kind of like the differences between the two of them. They are located at two different kind of depths. Kaneohe is the one offshore is around 15 meters deep. And the CREM one, which is the Coral Reef Monitoring Platform, that's how it stands for, is around 3 meters depth. So what we want to do is compare the diurnal scale variability between the two of them and investigate any changes in diurnal amplitudes over time and see if they're changing, and identifying and constrain the seawater CO2 changes during these diurnal variations or cycle. This is how the time series looked like. I'm showing here a time series from 2008 to 2023 of seawater PCO2. The blue dots are kind of the raw data every 3-hour measurements, and the solid lines are monthly means. And we have, this is the challenging of like analyzing time series is the challenging of missing data. But however, you can see that there is a nice seasonal cycle, multi-annual patterns, and even at this time scales and this time frequency is our time domain. It's difficult to see the diurnal variability, but once we move to the frequency domain, then we can start like kind of decomposing the signal. So what I did, or what I'm showing here, is the one-size spectral density for, one moment. Okay. The one-side spectral density for CREM and Kaneohe for the PCO2 seawater. And we can see here easily that there is 2 power spectral, or kind of like which one is more dominant, where CREM is the more dominant one, and the Kaneohe Bay have less kind of like a signal. And we can identify also 3 major signals, one at 26 to 24 hours peak. Those peaks are related to day and night processes. We can also identify 15 and 12-hour processes kind of related to tidal changes and semi-diurnal cycles. And 8-hour peak, which was still not clear about what is this mean, but is clear in the reef side and not at the offshore side. We speculating that this is due to different kind of like maybe more localized short-term cycles, like maybe irradiance, wind patterns, short-term temperature changes. But that's there. I want to show you to you, and then We also, with this type of data, which I am mentioning is every 3 hours, we cannot understand any processes that are less than 6 hours because those signals would be alias to other signals. So to start decomposing the signals and seeing any variations over time and see the differences between the two, here I'm showing range, kind of the maximum minus the minimum of the PCO of the different parameters. Here I'm showing you temperature and salinity. Just to the takeaway here is that the reef side is in gray and the orange side is the offshore Kaneohe mooring. And here, takeaway message is that we can see that temperature and salinity is a little bit more amplified at the reef side. That may be because of the depth and where they are located. However, they're not that different compared to the PCO2 ranges. And this can this show us kind of like how extreme daily fluctuations are experienced at the reef side. Around more, the range at this side is in average about 200 microatmospheres. And that could experience that during a diurnal cycle. And to further see if these variations or diurnal changes are changing over time, I'm showing you here the standard deviation of the diurnal changes for temperature and salinity for both reef, CRIM in gray, and Kaneohe offshore mooring in orange. And we cannot see any patterns of increasing or decreasing. They're pretty steady. What we can see is that CRIM, again, have larger diurnal variability. But when we go to see PCO2 in seawater, we can start seeing trends. Oops. Oh, that didn't show up, but there is a linear trend on PCO2. And even though it's small, we still, it is significant, around 0.06 microatmospheres per decade. And now we're trying to like understand or asking questions about is a biological change or this is a cumulative long-term OA trend showing this in the diurnal cycle. Here, pretty briefly, this is the PCO2 in seawater at every hour. For example, at midday, at 3 a.m., at midnight, sorry, 3 a.m., 6 a.m., 9, 12, 15 to 21. And we can start like looking at where is this variation come from? Is there any particular time where we see changes in the diurnal variability? We see trends in some of them that are significant, but I wanna just focus on that the less variability is shown from 3 a.m. to 6 a.m., which is coinciding to the peak of respiration, that's what we think. And most of the variability occurs from 3 p.m. to 6 p.m. And that is also coinciding with the gasification and photosynthesis. And when we looked at the oxygen as our proxy for net ecosystem production, we can confirm that that is the case where we see photosynthesis at 3 to 6 here in the positive values, and respiration during the night from 9 p.m. to 6 a.m. And we can also see that this net cumulative production is changing over time, where we can like maybe see an increase through time on net ecosystem production. And the units of this is millimole per meter square per day. I'm happy to discuss more about this, showing really quick. This is a seasonal cycle for net ecosystem production too. And we can see that there is spring, and during the summer month, there is more photosynthesis, or net ecosystem production is higher than during the fall here. And there is also changes, interannual changes. But looking at, we still need to more understanding on on the inorganic carbon cycle, and we still, are in the process to develop the methods to see if we can use the buoy data to constrain that part of the carbon cycle. But using the discrete measurements that we have for both sites, we can identify or have an overview of the major metabolic processes here using dissolved inorganic carbon and total alkalinity. And here we can see that calcification is the major dominant process for the Reef side compared to the Kaneohe side, which is now in purple. So calcification, take away from here is calcification can decrease total alkalinity by about 200 micromoles per kilogram compared to the offshore side, which is around 50 micromoles per kilogram. So to conclude, some of the remarks is that the MAP CO2 buoy at the Reef side have a highly dynamic diurnal scale variation in carbon chemistry relative to the Kaneohe buoy or the offshore one. Calcification and respiration are the dominant drivers, contributing to these extreme diurnal variations at the Reef. And we have the secondary frequencies, such as the 12-hour diurnal or semi-diurnal tidal cycle and 8-hour variations that we still are looking to see what it is. Can influence PCO2, but the major metabolic processes are the primary dominant drivers for PCO2. And we see an increase in the diurnal variability at the PCO2 parameter. And we're still emphasizing or trying to understand if this is a biological increase on some of the metabolic processes or if it's a long-term trend due to ocean acidification. So thank you so much. And that's it.
Thank you very much, Melissa, for that great presentation and for sharing with us your work on carbonate diurnal frequencies at your study sites. Our next speaker is Maisy Lus. Maisy earned a Bachelor of Science in Chemistry from the University of Papua New Guinea in November 2020. She comes from the beautiful island country of Papua New Guinea, PNG, known for its abundance in resources, extremely high biodiversity, and its extraordinary cultural diversity. PNG's remarkable diversity is indisputable in its oceans as well, filled with incredible life and mystery. In 2020, Maisy successfully incorporated a local NGO called the Fish Reef Project, PNG, with a vision to see ocean life thrive in her country through the creation and placements of man-made reef units. She was awarded a GOA-ON in a box kit and is implementing ocean acidification monitoring into the Fish Reef Project. Maisy has been selected as a Jimlin Scholar for the 2024 to 2025 cohort. Congratulations, Maisy. She is about to pursue a master's degree in applied marine science at the University of Plymouth in the UK. Maisy, the floor is yours.
Good afternoon all, and thank you, Luia, for the introduction. Apologies, I'm not sure if my camera is not working, so I'll probably have to go off camera for this presentation. But first of all, I'll just try to see if I can share my screen. not sure if you can see my screen. Can you see my screen?
Yep, all good.
Okay. First of all, I want to take this opportunity to thank the committee for inviting me to my presentation today. As they introduced me, my name is Maisy, and I am a current Go in the Box kit recipient from Papua New Guinea. Basically, for my presentation today, I'll just give a bit of my experience as a kit recipient and just share a bit about some of the challenges and lessons learned along my journey in trying to establish a baseline ocean acidification monitoring program in my country. So just a bit of background on Papua New Guinea. So Papua New Guinea is located in the Pacific, and we share our borders with Australia and Indonesia. Because we're located in the Coral Triangle, we're home to 76% of the world's coral species and over 3,000 species of fish. And we have key marine biodiversity hotspots located in Kimbe Bay and Melendez Bay provinces in my country. So the OA work that I do in Papua New Guinea is basically located in Port Moresby, the capital city. So most of my proposed assembling sites will be along the Motuan coastline in Port Moresby and central provinces. So just a bit of background on the Go in the Box and how I got involved in the program. So as mentioned by our moderator, I was successfully selected as a recipient for the Go in the Box kit in 2022. So this initiative was initiated by the Ocean Foundation in collaboration with the GOA-ON network to address ocean acidification. And I love this initiative because it was aimed at filling the ocean acidification monitoring gaps in the world, specifically in underrepresented regions like the Pacific Islands. And I'm fortunate to be a recipient of this kit. Yes, in partnership with the GOA-ON network, the Ocean Foundation were able to issue different kits to other recipients in the Pacific Islands as well. And this was sponsored under the NOAA Ocean Acidification Program, Fisheries Oceans Canada, and with support from the Pacific Islands Ocean Acidification Center, I was able to receive in-person training in Fiji and also online trainings through UNESCO. So I like this initiative because it's really aimed at equipping and training locals like myself to enable long-term and cost-effective data collection concerning ocean acidification, ultimately like contribute to the SDG 14 Life Below Water and International Climate Action Goals. So I'm very grateful for the Ocean Foundation because of this. So basically the Go in the Box kit contains a range of different ocean acidification equipment, measuring equipment. So below you can see a couple of images of the kit that I received in Papua New Guinea. So I was awarded the kit in 2022, but the kit arrived this year in April. And you can just see them unpacking it from the containers, and everything arrived as listed. So the kit includes sensors for in situ measurements, ocean acidification measurements using the sunburst ISM pH sensor, and you also have the different pressure sensors, the CTD diver, and also other related lab equipment for measuring total alkalinity and spectral photometric pH from discrete water samples. So it also came with a range of other glassware and laboratory related equipment to support with just basic ocean acidification monitoring. So upon receiving the kit, I first tested out the kit in April this year on a research expedition in collaboration with KAUS, the King Abdullah University of Science Technology, through my peer-to-peer mentor as part of the peer-to-peer mentorship program. So during this expedition in April this year, the expedition was aimed at, the expedition was in Kimbe, Papua New Guinea. It was aimed at unraveling coral reef resilience to global change using a natural extreme environment. So the natural extreme environment in this particular location in Papua New Guinea was a hydrothermal vent located on an island called Narage. And I tried to use that expedition as an opportunity to use the iSemis and the different pace sensors that I received from the GOA-ON in the box kit. And you can see in the picture there where I tried to test out my sensor. So here's just an image of some of the KAUS researchers along the hydrothermal vents underwater, just sampling some of the CCA and at around 30 meters depth. And the different sampling we were trying to carry out there. So here's just a picture of the iSemi sensor that I deployed. And in the next couple of slides, I'll just discuss some of the challenges and lessons learned from my first initial deployment. So from this image, you can see that when I deployed the sensor, it was deployed at a depth of 2 meters, approximately 50 meters from an active hydrothermal vent. Because of safety and other related reasons, we placed the sensor here. And the main challenges that I faced when deploying the sensor here was basically trying to come up with the correct frame with this like appropriate size to fit the sensor in the corner. And then I was grateful because when I was on board the ship, we had very supportive researchers that were experts in building specialized frame for different oceanographic sensors. So they were able to help me create a frame for mine. So I placed my iSemi sensor in one of those frames that we created in other oceanographic sensors as well on the same frame. And we were able to deploy this at the site. So that was one of the challenges I faced, just building the frame itself. And another challenge involved actually attaching the sensor on the frame so it doesn't move around. So yeah, just finding the right materials for that, especially when you're in a rural area away from the city without anything. You have to make sure that you have all your ropes and everything that you need to connect your sensors. Other environmental challenges at the specific site where we tried to deploy them, where we had issues with wave energy and having large coral boulders around the site area. So just environmental challenges with putting the frame, navigating the boat along the coral reefs, and then having someone transport the frame from the boat to the actual site. So you have, yeah, environmental challenges with the site itself. And then you have another challenge that I faced was the data retrieval issues. So the sensor was deployed at that specific site for approximately 3 days. And during the data retrieval, when we retrieve the iSemi and try to offload the data onto the computer. The data was offloaded successfully, but then it started reading negative numbers, and I was confused when I saw the reading. So I was thinking maybe because the sensor might have, maybe some air bubbles might have been introduced into the system, or there's still a bit of troubleshooting that I have to do to figure out why. After 3 days of being deployed, even though it was deployed successfully, when we retrieved it, the data ended up missing some figures. Because it was sampling every hour and then it ended up missing figures. So I still have to figure out why the specific sensor did that. So that was one of the major challenges that we faced during that specific deployment. And I was happy it happened in the presence of other experts as well on the boat. So they were able to help me troubleshoot that issue. And hopefully during my next deployment, when deploying the sensor out at sea, we should be able to make sure that all air bubbles or anything of that kind is not present when deploying and retrieving it. And also the sensor can be very delicate, so you have to be careful when switching bags. There's a specific salinity bag that always comes with the sensor that you have to have attached when retrieving it. So having that attached and detached correctly as well is another key to making sure that you don't ruin the data that you collect. So those are some of the challenges that I faced with that initial deployment. This is just a picture of what the raw data looked like when I offloaded it onto the computer. So you can see in the fourth column, all the data came out negative instead of a perfect set of data. So it was sampling hourly. At that specific site, we had salinity at 35 parts per million. Temperature, because it was at the hydrothermal vent, you can see the temperature was at 32 degrees Celsius. It was pretty hot water where the sensor was in. It was just the pH readings that came out negative numbers. So that's something that I still have to troubleshoot when I return back to Papua New Guinea. So yeah, so these are just some of, based on my experience from deploying that sensor, this is just my recommendations for anyone. If you're listening, based on my experience, it's better to deploy the sensor when you have more than one person or 4 people. It's better to have a team of people out with you to support you when you're deploying this type of equipment. Because when we were deploying this, we needed someone to help offload it from the boat, and then you need someone to put weights on it when it's underwater. And if you're collecting discrete water samples as well for your alkalinity testing, you need someone collecting water samples as well. So there are a lot of parts moving during these deployments. So it's better to work together as a team and have people that are trained to do that. And preparation is also key as well, especially if you're offloading data. You need to have your personal computer with you on a boat, if you're on a boat, or nearby for you to transfer data as soon as possible. And yeah, choose sites that are more calmer and easily accessible so you don't end up damaging equipment or hurting yourself. And yeah, safety is always key during these deployments. So post-expedition, so after the expedition, I came back to Port Moresby, and I was given two sensors. So with the other sensor that was still working, I engaged a local to build a frame for me. So he built it out of aluminium. And then I was able to attach my sensors onto the frame that I designed. And apart from that, I, with the rest of the components of the kit, every weekend I usually take out students and other interested people in the community that just want to learn how to use a simple compass or a GPS, or just training them how to use the different water, the discrete sampling bottles, and just how to collect alkalinity samples and take sea surface temperatures and all that. So yeah, that's the ultimate goal. Currently studying here in the UK. So when I return back to Papua New Guinea, this is what my work will look like. Continuously going out in the field, aim to train more Papua New Guineans in this area. And hopefully I'm able to have this deployed at the sites that I have in mind. And then, yeah, continue to train more people to use this based on the lessons that I've learned. So yeah, these are just my future plans. So I'll be finishing studies here in 2025, and yeah, just continue to use the GOA-ON kit to collect just simple baseline monitoring in our coral reefs, seagrass beds, and other coastal environments, and collaborate with local stakeholders to build capacity, because I believe that's what we really need in the Pacific, more capacity building. And yes, just contribute our findings and contribute to the overall SDG 14 goal. But yeah, that's the end of my presentation, and I'd like to acknowledge all the Ocean Foundation, NOAA, the Fisheries, the Oceans Canada, the peer-to-peer mentorship program, even the Pacific Islands Ocean Acidification Center under Dr. Katie Soapi, and we have Azaria and the team there, and also UNESCO and my collaborators from KAUST. That's all. Thank you.
Thank you very much, Maisy. Thank you, Maisy, for the wonderful presentation and for sharing your work and also the challenges of the OA monitoring in PNG. We are excited about the OA kits, and we are keen to hear more about your work in the future. Our next speaker needs not much introduction. She is Azaria Pickering. Azaria is an ocean monitoring technical officer with SPC in Suva in Fiji. She works with the Pacific Islands Ocean Acidification Center, supporting colleagues throughout the Pacific as they implement OA monitoring programs. Azaria manages an OA observing program in Fiji. Azaria, the floor is yours. Thank you.
Thank you, Luia, for that introduction. So I'll be presenting on behalf of Dr. Katie Soapi on ocean acidification from a regional perspective. Next slide, please. So this is a map of the Blue Pacific, where we are 22 countries and territories with over 10 million people serving as custodians and stewards of about 20% of the world's exclusive economic zones. We have limited capacity and resources in the Pacific, and so we pull in our resources and our expertise and shared goals to maximize the impacts of our efforts. And this speaks to the Blue Pacific narrative, one shared ocean identity, ocean geography, and ocean resources. Next slide, please. So why is ocean acidification a big issue for us? As you just saw, we are more ocean than land, and so our livelihoods depend on thriving marine ecosystems, but also because majority of our population reside along the coastlines. So looking at global projections and understanding what They mean for us here in the region, globally, our oceans are 30% more acidic compared to 200 years ago. And downscaling that to the Pacific, what does it mean in terms of vulnerability? So this means that in terms of impacts, our livelihoods are affected, food security, income, cultural practices, as well as health and well-being. And the reality here in the Pacific is that we lack data, we lack the capacity and the infrastructure to understand ocean acidification and what's happening in our oceans. So, we really need the support and investment in monitoring of our oceans so that we can make evidence-based decisions and inform policies. Next slide, please. And I just wanna highlight this study, the Pacific Islands OA Vulnerability Assessment. So this study provides a lot of useful information for us to be aware of, like identifying key hotspots, the vulnerable components to ocean acidification, like coral reefs, shellfish, as well as contributing sources to OA, and implications for monitoring, policy and planning. And it also highlights the need for informed pilot projects for local adaptation. Next slide, please. So one of the initiatives fulfilling the need for the informed pilot projects is the Pacific Islands Ocean Acidification Center. And before the center was established back in 2017 and 2018, there were capacity building workshops being held for beginner and advanced levels. And also there were 8 Pacific Island countries that received GOA-ON in a box kits. However, there was no regional coordination. And so this is where the center came together to serve as the hub to provide that support and to provide the services needed for monitoring programs. So in 2021, the center was established. And since then, there have been a number of trainings held both online and face-to-face. And currently, the center plans to have country visits in the future where experts travel in country to provide training and support for setting up monitoring programs. And it's important to know that this is what we specifically know for the Pacific Islands Ocean Acidification Center. There are other initiatives and projects that are taking place, being carried out in the region, and that I'll highlight those in the slides to come. Next slide, please. So the center was initiated through combined international and localized efforts to support ocean acidification research and monitoring in the region. And this is achieved through training, through the distribution of equipment, assistance with OA data management, and also providing access to international networks that support monitoring, action, and policy efforts. And to the right is a timeline of the kit distribution, the GOA-ON kit distribution in the Pacific. And so these are the countries that have received the kit. Next slide, please. Looking at the broader PI-TOA region, these are some of the initiatives, like some of the projects being carried out. We have the Palau International Coral Reef Center through partnership with IAEA and KIOST and SPREP. And this project is supported through NOAA. And so they're leading national efforts to understand and respond to ocean acidification. So they have a lab, they also have a monitoring buoy, and they also have a GOA-ON in a box kit. We also have a joint project between KIOST and SPREP that supports the Samoa Met Office. And this is also an institution that that the center collaborates with. In FSM, they have a MAP CO2 system that's deployed in Chuk, I believe. And this is through partnership with the NOAA's Female Carbon Group and KIOST. And we also have national-wide programs in the larger Pacific, like we have New Zealand's OA Observing Network that's supported by NIWA. We have CSIRO's OA monitoring and other platforms in Australia. And we also have the Hawaii Ocean Time Series that's supported by NOAA's Female Program. And throughout the region, we also have OA buoys that are deployed around the Pacific that NOAA is actively participating in. Next slide, please. So this map highlights some of the initiatives that I just mentioned, who's collecting data and the types of methods that they're using. Some are just starting, and some of the programs have stopped. So this is a working map, and we add information as we get them. And you can imagine that there are many actors, and most of the work is done in silos. Because most of this is conducted by different researchers, different organizations. And so this is where collaboration and coordination is important, because that allows us to leverage off each other's capabilities as one Blue Pacific. Next slide, please. And these are some of the activities the Pacific Islands Ocean Acidification Center has taken part in or has led to continue the OA efforts in the region. We've had the hands-on training with 9 Pacific Island countries, also the sensor training for the pH sensors that are part of the GOA-ON in a box kit. And Fiji, as part of the center, also established a monitoring program last year in February and is currently active. And also they've been able to establish a network with other Pacific Island countries and their collaborating institutions. Next slide, please. So this, in terms of partnership, the Pacific Islands Ocean Acidification Center is in partnership with USP, SPC, University of Otago, and MIWA. And this is through the support of the Ocean Foundation and NOAA. And there are other collaborating institutions like SPREP. We also have the Pacific Islands Marine and Ocean Services, PMOS, that's under the Pacific Med Council. Also New Zealand's Pacific Partnership on Ocean Acidification, the NZPBOA project. And that was a collaborative effort between SPREP, USP, and SPC to build resilience to OA in Pacific Island communities. That project was in Samoa and Tokelau, I believe. And then we also have countries in partnership with KIOST that are actively carrying out OA work in the region, in Samoa, in FSM and Palau. And then there's also other partnerships with NOAA and the Ocean Foundation and other organizations that are carrying out OA projects in the region. And yeah, that's the end of my presentation. Thank you.
Great, thank you, Azaria. Thank you for your presentation on the OA monitoring from the Pacific perspective. I also acknowledge your work to support the PI-TOA hub group. So thank you. Our last speaker is Ms. Caitlin Lauder. Caitlin is a program manager at the Ocean Foundation, supporting the Ocean Science Equity Initiative. Most of Caitlin's work revolves around advancing ocean science capacity globally, particularly for ocean acidification monitoring. As a marine biologist, she has researched the effects of ocean acidification and ocean warming on ecologically important, on economically important crustaceans, including California spiny lobsters. Caitlin, the floor is yours. Thank you.
Wonderful. Thank you so very much. I'm really honored to join this group. I don't live in the Pacific Islands region myself, so I'm especially grateful to be joining you all, although I do live along the Pacific coast, so we're connected in that way. I'm going to share my screen, both the sides, and we'll get going. All right, so as you can see, the work that I'm sharing here today is not just my own, but through the work of a number of different authors. I'm going to be talking about some accessible practices and guidance for ocean acidification monitoring that I think is helpful for this region and other users as well. You'll see some familiar names of some of my co-panelists like Melissa and Azaria here as well. So Maisy provided a wonderful introduction to the GOA-ON in a Box kit. Thank you for that, Maisy. I won't touch too much on that, but this is a suite of equipment to sample and measure total alkalinity, spectrophotometric pH, and some sensors. If you're interested in learning more about that, you can go to the GOA-ON website and get a link to the spreadsheet where we maintain our GOA-ON in a Box kit list. But our work with the GOA-ON in a Box kit and distributing around the globe, including the Pacific Islands region, has really led us at the Ocean Foundation and a number of our partners to find additional resources to complement the use of that kit. There are some available resources online. This is not a comprehensive list that you see here on screen, but manufacturers have sometimes produced guides for their instrumentation, such as sunburst sensors here, with their manuals and client software to support the ICME. I think it's an important hallmark for low-cost equipment and technology developers to include guides like this because it makes their products so much more accessible. And then among our community, we've also created some great resources for use of the kit and oceanification monitoring in general. At the Ocean Foundation, we've produced 4 videos that are available in a couple different languages on our YouTube channel. There's also the OA Information Exchange to share tips and tricks, other online guides and videos like what Remarco has produced in the Latin American Caribbean region, information on the SCG 14.3.1 portal to submit your data, which I understand is open right now, and then classic resources like the Guide to Best Practices. But we're really looking for a one-stop shop that's practical for researchers to be able to work through research methodologies. And that led to our work to create a resource that's tailor-made for the equipment and the chemicals provided in the GOA-ON in a Box kit that matches the knowledge of new kit users. Our group effort started a number of years ago. You have probably heard about our work on the practical best practices if you've attended OA weeks in the past. But what's super exciting is we finally published this resource online. We have an online platform on a website called PubPub. So if you go to oceanacidificationcookbook.pubpub.org, you can see it there. I'll also note that webpage may be changing in the future, but we have a DOI, which I will pop in the chat. I don't know if I have access actually to all attendees. Just keep a look at that DOI on the recording, and you'll be able to always access the practical best practices. Just for an example of what are in all these different guides, of which there are more than 20 on our website, we have links to other best practices. So the big, thick manuals are in there for more information. We cross-link to other guides within the practical best practices, so it's easy to jump between related practices. We link different version or language options when available, particularly for videos. Videos that describe and showcase in these methodologies are directly linked in the web page, which is really spiffy. And then following this introductory section, all of our guides have a step-by-step section that enables researchers to follow along and get all the tips and tricks that are really useful for conducting ocean acidification monitoring to weather quality standards. Also, for some of the guides, we have associated spreadsheet templates. So if there's an Excel file that's useful for conducting data analysis or using CO2 sys, those are directly embedded in the web pages. And then we also have plenty of images to complement the textual information and provide hopefully a really solid example or guide for conducting these methodologies. So I'm going to turn now to that web page so we can take a look together. So following the introductory section, here's also a link to the Go in a Box kit list right there. We have a couple different sections that divide up our documents. The first is a set of general practices, which I think is really useful not only for GOA-ON in a Box kit users, but anyone who's getting started in the ocean acidification field. We have some really nice guidelines for laboratory management that talk about safety, but also efficiently using resources, which is really helpful when they may be limited in the region that you're working in. There's a nice document on selecting a research site with particular considerations for initiating ocean acidification monitoring. There's a very nice checklist for field sampling. So as Maisy was mentioning, all these little bits and pieces that are really important to bring out to the field, everything from zip ties to the specialized equipment for monitoring can be hard to remember. So we put together a nice little checklist for that. We have a guide on cleaning sample bottles, whether you have access to specialized soap or just deionized water, or if you're using your bottles for biological sampling, there's some guidance in there too. And then a nice little spreadsheet template for information that should be collected while out in the field that will enable metadata submission for the SDG 14.3.1 data later on. Very handy. The next section is on sensors, which is more particular to the GOA-ON in a Box kit, but we have a guide for using the sunburst sensors, ICME, a set of frequently asked questions. So Maisy, again, as you perhaps figure out some of these issues that you're experiencing, or we can work with some experts there, we can pop that little bit of learning into this FAQ guide to help other researchers that are working with the ICME sensor, and then also a user guide for the CTD diver that's included in the kits. For our discrete samples section, we have a nice guide on collecting bottle samples, which I think is again useful for many different researchers, using the modular total alkalinity system for included in the kit, preparing the pH indicator dye solution, the metacresol purple, going through the process of using a spectrophotometer to measure pH, and then getting into a little bit more of some back-end guidance. So assessing the quality of measurements with reference materials, whether that's Tris Buffer or the nice reference materials from the Dixon lab or perhaps other laboratories in the future. How to process mercuric chloride as a research waste product using a filtration kit, and then calculating the seawater carbonate chemistry system. In this case, with a walkthrough of using CO2 sys, this includes the Excel file as well as some worked examples too. And then in our last major section, we have some documents on data management. So some data quality control, quality analysis guidelines. It's very handy. And then a quick guide to submitting data for the SDG 14.3.1 indicator. And then our last document in here is a set of handy community resources. So I think this is another document that's widely applicable. We have links to other folks in the OA community who have put together guidance, different places to go to if you have questions on certain things, and just some really nice general resources. So I'll pause there and turn back to our set of slides and just note that we've got this version 1 out. We would love to hear your feedback. We are now working on a version 2 with even more guidance for those who are joining the ocean acidification field. We're envisioning a decision tree. So whether a researcher has a set of resources available and they're wanting to figure out what sort of research questions in the field that they can address, we hope to be able to provide them guidance through this decision tree. We're also thinking if a researcher has questions that they want to answer, we can help them assess what sort of resources are needed. What training should their personnel or their staff obtain? Where can they get equipment, or what sort of equipment might they need to answer those questions? We'll hopefully create a couple entry points to make it easier to dive right into some of the biggest gaps in our community. We're also putting together a compilation of biological resources. So for researchers who are interested in understanding the impacts of ocean acidification on marine organisms, or getting engaged in the fields, and then updating our version 1 documents as well. This is a call. If any of you say, that sounds amazing, feel free to send me an email. I'll put the link, or you'll see it on my last slide. We would love additional authors on this. If you have more ideas, if you would like to plug into some of these existing documents, I think we're quite a welcoming group. It's really fun to engage, and we love all sorts of perspectives, whether you've been working in the field for a number of years or whether you're coming from more of an entry point and can provide a really great perspective of what it's like to dive into OA research right now. So in another minute or two, I'm just going to touch really briefly on some guidance we've been working on at the Ocean Foundation in partnership with PIOAC on using mercuric chloride, because that's something we've been hearing about as a concern in the Pacific Islands region. In partnership with one of our former staff members, Madeline Warner, we were looking into how a couple of international conventions apply to mercuric chloride to think about importing mercury in terms of in the actual kits, how to safely dispose of mercuric chloride, and then make sure that we can provide guidance to those of you who are conducting this research. So I'm not going to go through all this different text. If you're watching the recording, you can pause it and read it. But essentially what we've learned through the Minamata Convention, which governs mercury, and the Basel Convention, which is looking at hazardous waste, mercuric chloride in the resin filters that are provided in the GOA-ON on a box kit can be classified as waste. And given where the Minamata Convention currently is now, because this is a research item and is being used for research purposes, there is some room in the text to interpret this waste as something that can be transported across international boundaries for the purposes of safe disposal. So while mercury typically is regulated in terms of moving across country borders, because this is a research waste item and a relatively small quantity at that, we believe that there are these safe disposal mechanisms. So in summary, just really briefly, the resin that contains mercuric chloride is a mercury added product under the definition of these conventions and a product of research. It is a research item, so it's excluded under some of the annexes of the Minamata Convention, and we are allowed to safely dispose of waste under that convention. So this super brief run through. If you have any more questions, I'd be happy to answer that. I will note that our staff capacity is a little bit low to continue this at the Ocean Foundation, but we're interested in continuing to support researchers who are bumping into challenges with conducting OA research because of this issue. And I'll also highlight some work that Azaria has done to create a document that really dives into some of the regulations and the documentation in a lot of countries in the Pacific Islands region and how it refers to mercuric chloride. So I might tag you over to Azaria for any particular questions. I just want to highlight her work here. With that, I'll say thank you. Here is my email if you're interested in taking a look or hitting me up about the practical best practices. So thank you.
Right, thank you. Thank you very much, Caitlin, for the useful information about the links that you shared on your talk. And that brings us to the end of the first half of our session today. Thank you to all of our speakers. We would now like to open the floor to questions from our audience. So you can either type your question into the question box. I think everyone knows where that is, at the bottom of the control panel. And on the right side of the screen, or you can raise your hand and we will call on you to ask your question directly. While our audience and everyone is getting their questions up, I have a couple of questions for Sebastian to start off with, if you don't mind, Sebastian.
Unfortunately, Sebastian's had to leave us.
Oh, right. Okay.
I will post his email in the chat for
yes.
future questions to email to Sebastian.
Okay. I have a question for Melissa, if that's all right, Melissa, while we're waiting for others to come up with their questions, if they have any.
Oops. Yes.
Yes. Hi, Melissa. Interesting work that you're doing there about the variability in the carbonate chemistry. You said that you take measurements every 3 hours. What is that a standard for that particular sensor that you're working on, or it's just your own method?
No, that's actually standard for all the MAP CO2 buoys that are located at different reef sites and around US continental waters.
Right.
So yeah, it's kind of like all of those buoys are measuring every 3 hours.
Right. That's interesting because we have a buoy here in Samoa with a meteorology office, not here at the university, but I wonder, I would like to know how often that they program their equipment to take these measurements. I also was interested to ask Melissa about the photosynthetic rates that you showed in your graph that you showed in your slides, please. So I don't know if you can show that graph again, but I was interested to know what's changed over the years that's made those peaks vary a little bit from each other over the years. Has it been less sedimentation or something like this in those areas?
Good question. It may be a change in the benthic communities. It could be changes in the water column communities. Science is a measure of the net community production. It cannot distinct between the benthic and the water column. So it's kind of hard to pinpoint where is this coming from. I guess that to understand that, we will have to look at other parameters, like for example, maybe some chlorophyll data, maybe looked at any other data sets that tell us a little bit more about the benthic communities, or if there is some other data sets available at the site. But yeah, with the chemistry data, it's kind of hard to pinpoint.
Yeah, thank you for that.
But good question.
Yeah, wish you all the best with your study. And is there any question from the audience? Yes. There's a question here for Maisy. And that's coming from Caitlin. Caitlin, do you want me to read your question or you want to ask?
I can read it. Maisy, you talked about working with the community members, which is really exciting. Can you share a little about your discussions of perceptions of oceanification, or concerns that they may have with this challenge, if that's something that you've discussed with them?
So initially, I started off the conversation about ocean acidification, but most of them aren't really aware of the concept of ocean acidification. So I tried to introduce the concept to them by making them help me do some measurements out at sea. So we collected some discrete water samples, and because they're mostly involved in mangrove rehabilitation and mostly to do with seagrass. I guess when communicating ocean acidification with them, I think they sort of leaned more into the idea of if they planted more mangroves or help preserve the seagrass, that would absorb the carbon dioxide and make the water less acidic. So that's basically their understanding of how ocean acidification is. So it's just me working between those two ideas that they have, and yeah, just keep taking them out more on site, and then just explaining to them more about ocean acidification so that they can fully understand the whole concept behind it. But yeah, they've been very receptive. They really like the capacity building side, being able to learn about new technology and the new equipments and all that. So just using that pull from the community has been very helpful from my end. Yes. I hope that answers the question.
Yes, thank you.
Thank you. Yeah, right, great. I have a question for Maisy as well, because you mentioned, Maisy, about the natural thermal vents and where you placed your ISAMI,
Yes.
um is that, and you also mentioned some challenges of negative numbers coming out from there. I wonder if the ISAMI is sensitive to the very large changes in the pH, or if it, because it's near the pH range where pH could be low, and maybe that makes the ISAMI a little bit more sensitive to that.
That's what I initially thought. But from when we were on the expedition, the team from KAUST also had an ISAMI with them, but their ISAMI was bigger. I think theirs was the ISAMI PCO2. So it measures pH and PCO2 at the same time. So so they had their sensor deployed next to mine as well, just in case if one of them doesn't work. And when they retrieved theirs, they actually got their pH readings from the site, and it was just mine that didn't come out. All the numbers came out negative. So it made me think maybe it might have been something with how we retrieve the sensor, or I think there's a standardized solution, like a bag that's connected to the sensor before you pull it out. Maybe that might have been I wasn't connected or disconnected correctly in some way. So yeah, I'm just still trying to figure out what went wrong. Because I have two iSemis, I don't want to spoil the only good one that I have. So I just really want to troubleshoot what really went wrong before deploying the only good one that I still have left.
Yeah. All right. Thank you, Maisy. I think you're doing a good job. So we look forward to hearing more update from you in the future, in the next SIP webinar. Is there any more questions? If there's no further questions, then I pass it back to Rachel to wrap up our session. Thank you, everyone.
Thank you, everyone. So we would like to thank the sponsors, organizers, moderators, and the speakers for a great session today. And thank you to the audience for joining us for this session and for any other sessions that you've attended at Ocean Acidification Week. Don't forget to consult the website and to register for any other sessions in the next coming days that might interest you. If you would like to stay up to date with the GOA-ON community, consider signing up as a GOA-ON member at www.goa-on.org. And there's an open call to join the GOA-ON Executive Council out at the moment, so please check that out. So just finally, thank you everybody once again, and we'll see you next time.